All right, so what I'm going to do is tell you about the cardiology research, give you a little bit of background for what got me interested in this, and go from there. So my interest has really been in trying to understand the genetic causes of congenital heart defects, and probably most of you may not be aware that it's by far the most common birth defect, affecting almost 1% of all newborn infants. And so even though it's by far the most common, and it's most likely to be caused usually from a genetic etiology, the specific gene that causes the heart defects is usually not known. So when I'm down at Rady Children's taking care of the 25 or 30 patients in the hospital at any time with congenital heart disease, in most cases we don't even know what we're treating in terms of the cause. And so my feeling has always been that by understanding the genetic causes, we can get a better handle on the mechanisms and hopefully come up with more effective strategies for treatment, and maybe even some preventive strategies, which I'll come back to in a couple minutes. And so this was the heart defect that I was actually originally and continue to be most interested in, something called hypoplastic left heart, which I know is more familiar to some of you than you ever wanted to be. It's a severe congenital heart defect in which basically, as you can see here in this cartoon, compared to the normal heart, the left-sided structures just don't develop normally. So that since the left ventricle is normally the ventricle that pumps blood out to the body, it's unable to sustain that, what we call systemic cardiac output, and so it's a uniformly fatal heart defect unless one of two things are done surgically, either a heart transplant, but that's not very feasible in most cases because there's such a shortage of donor hearts, or we do currently, most commonly, a three-stage palliation, surgical palliation, where we essentially convert the right side of the heart to do the work that the left side of the heart did, or is supposed to do. So I got interested in Jacobsen syndrome, for those that were here last night, initially back in the first month of my cardiology training here at UCSD, when I came across this little boy, Edgert, who basically had a form of hypoplastic left heart. In fact, he just had an echocardiogram the other day. He's probably coming up and going to need another operation very soon. And so, of course, I had never heard about Jacobsen syndrome back then. And this was a picture, actually, of his chromosomes back in the days when we did karyotype analysis as a first-line genetic test. And I think you can appreciate better than anyone what his problem was due to. As you see there, he was missing a chunk of that chromosome 11. And so for me, that was very compelling, suggesting that there must be a gene involved, or genes involved, at least in that missing part, that must be accountable for the heart defects that he has. And of course, my bigger interest is not only, my parallel interest is not just in what causes heart defects in Jacobsen syndrome, but how that might be relevant to the much broader, nearly 1% of the general population with congenital heart defects. And so that really set the wheels in motion, and through my fellowship and beyond, I really did everything I could to learn everything about the syndrome. As I mentioned to you, we had our first conference in San Diego in 1998, and that was kind of the beginning of this formal process to just sort of systematically learn as much as we can about all the aspects of Jacobsen syndrome, which I think you can appreciate. We are still learning to this day, some 15 years later. And so this is a table just from that paper in 2004 that began to give us some insights into the spectrum of problems that occur in Jacobsen syndrome. So obviously for this talk, I'm just going to focus on the cardiac defects. And there were a few things that were already apparent. One is that certainly not every child with Jacobsen syndrome has a heart defect. So roughly about half of them have heart defects. And if you take that half that do have heart defects, I kind of decided to divide them into two classes of defects. One class is the so-called left-sided heart defects. So the most severe case would be what I just showed you, hypoplastic left heart syndrome. The least severe case is something called a bicuspid aortic valve, which actually happens in about 1 to 2% of the general population, and then everything in between. And what was actually quite remarkable as I learned more about this was that compared to any other known genetic syndrome, hypoplastic left heart syndrome occurred at a higher frequency in Jacobsen syndrome than in any other known genetic syndrome. And so that was really a compelling reason for me to want to move forward to try to understand everything we can about that. Now, interestingly, like I said, that accounts for about 2/3 of the patients with heart defects, but the other third actually had a wide range of other heart defects, as I've shown listed here. And so, again, if I took you down to our service at Rady Children's, for those 25 to 30 patients patients that were taken care of every day with congenital heart disease, the vast majority of the the heart defects that occur in Jacobsen syndrome you would see down there as well, or the other way around. So really, what I thought is, based on all those findings, this is a really very interesting and potentially very powerful opportunity to try to learn a lot more about just, again, in Jacobsen syndrome, but how that might apply to the general population. So again, half of your children have heart defects. There's an unprecedented high frequency of hypoplastic left heart syndrome, and most of the major congenital heart defects that occur in the general population occur in Jacobsen syndrome. So before I tell you about the research, from a practical standpoint, I wanted to just kind of share with you what our recommendations are in terms of what do we do when we have or see a child with Jacobsen syndrome for the first time. So a baseline evaluation should always include a imaging study, most commonly an echocardiogram, to define the cardiac structure and function. And obviously there are certain things that we may not be able to appreciate by just doing a conventional history and physical that you can only identify by an echocardiogram. And then we recommend that follow-up is kind of, well, it's obviously going to be dependent upon those findings. And by the way, these days we can do fetal echocardiograms as early as middle of the second trimester, so you can have a pretty good idea what to expect even before the baby is born. And subsequent follow-up really kind of depends upon what the results are of those initial findings, or if anything should change clinically, like the development of a new murmur, then for sure that warrants further follow-up. So in most cases, I think we can say that if your baby has a normal echocardiogram after birth, that it's very unlikely that there's ever going to be any problem down the road. Although I can't say that 100%, but with a fair amount of confidence now. Okay, so coming back now to the research approach, one of the things that was very clear from Laura Penny's paper that I described to you on Monday night was that there was actually a lot of variability in the size of the deletions. And so what we wanted to do is try to take advantage of that, to find the common region that's deleted in all patients that have a congenital heart defect. And what really sort of launched or accelerated our progress was the implementation of the array CGH that you've heard a lot about. And that then allows us to get really a single gene readout to identify what potential genes might be missing in association with the congenital heart defects. And so over the years, some of you have seen this slide from before, we went from Edgar's initial deletion, that's about 16 megabases, which means there are about 150 or more known genes in that region, through various patients whittling that region down to maybe no bigger than a region containing about 6 genes. And so this is just sort of a cartoon picture showing you with the black is just kind of the end of 11q, and the blue line that you see there is the smallest deletion going out to the end in a patient with Jacobsen syndrome that has hypoplastic left heart syndrome. So that deletion was about 7 megabases, so about half the size of the original. So that maybe took us down to a region containing about 50 to 75 genes, which is obviously great progress, but still pretty prohibitive in terms of trying to identify a single gene or maybe a few genes in there. So what really helped us was, thanks to the array CGH technology, we were then able to identify or come across some patients that had the same heart defects and other clinical findings in Jacobsen syndrome, but instead of having deletions going to the end, they have these so-called deletions within the chromosome, or what we call interstitial deletions. And I think what you can appreciate here is that the hypothesis was that if we look at the smallest region of overlap between the blue terminal deletion and these red interstitial deletions, that might help us to narrow down even further the culprits potentially. And so without going into detail, I'm just going to tell you that we were able to narrow that region down from what we started out to be probably over 150 genes, now down to about 6 genes, and all of a sudden that became much more plausible in terms of being able to study those individual genes to see what they might be doing in terms of heart development. And so I'm just going to tell you about the first gene, because that is actually the order on the chromosome, and it turns out to be the gene. It's a gene called ETS-1, which is a so-called transcription factor. It was actually originally identified in white blood cells and was known from a lot of groups studying it to have a critical role in the normal development of white blood cells, and also actually in their normal function, meaning in the immune system, which you'll hear about later on today, and also in pathologic states, for example, various leukemias in which ETS-1 expression is abnormal, and that causes certain forms of malignancy. So, as I said, ETS-1 is a transcription factor. This is just kind of summarizing what I already told you, but in terms of its role in heart development, nothing was known about the role of ETS-1 in the heart development in mammals or let alone humans. Now, we do know, of course, that transcription factors are a group of genes or factors encoded by genes that are critical for a lot of biological processes, including organ development. And in fact, the small number of genes that had been previously identified to cause human congenital heart defects in most cases actually encode transcription factors, so that made it, again, a very plausible candidate gene at that point. Now, having said that, it turns out that there had been some very elegant studies by a group at Berkeley at the time in an organism that actually lives right out here in the ocean called the sea squirt. And it turns out the sea squirt actually has a heart, even though it's not quite known what it's considered, I guess, as an invertebrate animal. But the sea squirt, this is actually a picture from a paper that was published in 1900, but it has this tube that actually functions as a heart. And that tube is a single cell layer that actually pumps the counterpart or the version of blood through the body of that organism. And if you look at it in real time with the right kind of photography, you can actually watch this heart beating. And so that's how it works, to propel the blood to the body. And so, again, this group from Cal Berkeley had been studying the sea squirt, and in particular the role of this ETS-1 gene in heart development. What I'm showing you here is actually these two red dots correspond to the two cells in the sea squirt that eventually give rise to that heart that I just showed you. And similarly, these two cells come from a common progenitor, but instead of giving rise to the heart muscle, they actually give rise to the tail muscle. So they come from a common progenitor cell and then diverge. But the nice thing in the sea squirt is, because it's a transparent organism early on, they can actually follow what happens as these two cells go on to develop into the mature heart. And they can also actually use that organism to define the actual pathways that the ETS-1 gene in the sea squirt is regulating. And I don't want to bog you down with too much detail, other than to tell you that what these investigators found was that there are two critical processes in heart development that were regulated directly by ETS-1. One is how those two cells that I showed you actually differentiate into a mature functioning heart. And as you can imagine, those cells don't just sit there, they actually have to move and get to the right place at the right time to form that heart. And what was quite compelling is, as we know with transcription factors, they tend to regulate a whole pathway of genes, and these investigators were able to identify some of the members of those pathways, and they actually turned out to be genes that were already known to cause human congenital heart defects. So this was actually a very gratifying observation to me to suggest that ETS-1 in heart development is probably a very widely conserved, highly conserved process. It's not something that's very esoteric and unique just to Jacobsen syndrome, but probably has a very central fundamental role in the biology of heart development, from anywhere from humans to sea squirts. So one of the things that you want to do when you study the role of a gene in any organ development is to try to actually figure out more about its function, in particular where is it functioning and when. So you don't have to get bogged down on this slide either, other than to show you that what we could do is look at where this gene is being expressed and to determine exactly which cell types in the developing heart is this gene being expressed. So, and it turns out the heart is sort of a mishmash of a lot of different cellular lineages, some from within, some start from other locations in the developing embryo, that have to be in an incredibly coordinated way regulated to come together to form the developing heart. And so what we found is that this ETS-1 gene is actually, to our relief, expressed in two very critical cellular lineages involved in heart development. One is called the neural crest, or actually the cardiac neural crest, and the other one is the endocardium. And so basically the rest of my talk, I'm going to tell you about what we think is going on for how ETS-1 regulates cardiac neural crest cell function during heart development. Don't worry about this slide. This is just another confirmation of isolated cardiac neural crest cells, just confirming again that ETS-1 is actually expressed in those cardiac neural crest cells. In this case, this is from mouse, which is where most of the rest of the studies are done that I'm going to tell you about. So one of the things to answer the question of whether or not a gene has a critical function in any kind of biological process is to ask the question, what happens when you take away its function? And so because there were already people studying ETS-1 for its role in lymphocyte development, we were actually to save about 3 years worth of work by getting targeted mice in which that specific ETS-1 gene had already been deleted. And so what this shows you is, compared to the left side of the screen, these are hearts in the middle and the right from 2 mice that had this ETS-1 gene eliminated from the beginning of conception, so from the beginning of the egg, and in all the tissues then. So when you take it out from the get-go, so what we call a global knockout, in a very specific strain of mouse, what we call C57B6, what we see here, and I think you can appreciate, is very clearly these mice have a hole or a gap here in this wall that separates the 2 ventricles. So we call that a ventricular septal defect, and I know that's actually the most common heart defect that we see in Jacobsen syndrome. And so that was very gratifying, suggesting that at least in some ways we can sort of recapitulate in the mouse what we know is going on in the humans. So we can use some other imaging modalities that some of your children may have had. This is a CT angiogram of a mouse embryo, and again, just to orient you, that's the left ventricle, that's the right ventricle. You can see the wall, then you see this gap, so that's the ventricular septal defect. And actually, just like we do with our human patients, we can do cardiac MRIs to create a 3-dimensional reconstruction. This white line here represents 1 millimeter, so this is from a mouse embryonic heart that's altogether only about 4 millimeters in diameter. And again, compared to the normal, what you can see here, here's the left ventricle. This is blood essentially going across this gap, the VST, and you can see here the right ventricle. And the other thing that you see here is that the position of the aorta is actually shifted over to line up more with the right ventricle instead of its normal location hooked up with the left ventricle. So we actually call that double outlet right ventricle, which really is characterized by this VSD and then this sort of malpositioning of the aorta. And again, let me re-emphasize the fact that this is what we see 100% of the time in this one genetic strain of mouse called the C57B6 strain. And interestingly, what we found is that if we look at some other strains, we see other heart defects. So kind of like your children, it depends upon this so-called genetic background what kind of heart defect you might see. And this is just to summarize that in this table. So in that background I just told you, 100% of the time, really without exception, we see the VSD and double outlet right ventricle. In a different background, we only see a heart defect about half of the time, and in that situation, the defects are actually variable. And then actually in a different background, the heart is completely normal. And to me, that's a very, very interesting observation, because it might help us to answer the question why it is that your children, you could have 2 of your children in which the deletion is identical, and in one case, like Amy, who was born with essentially a normal heart, and then Elliot, whose deletion is about the same size as Amy, required open heart surgery in the first week of life. So what it means is that we think that deleting the ETS-1 gene obviously is necessary to cause the heart defects, but there must be some other factors that can contribute to actually whether or not that phenotype is manifested. So we call that penetrance. So I want to come back and tell you a little bit more about this cardiac neural crest cell population. So as I mentioned, as the heart is developing, some of the cells develop from within that tissue. But in the case of the cardiac neural crest, it's actually a population of cells that originates in the developing central nervous system. And this is just a cartoon showing the specific location of that cardiac neural crest population of cells. And I think you can sort of appreciate that over time, during the development of the embryo, these cells have to migrate to get to very specific structures of the developing heart. And you can imagine that if anything goes wrong to disturb that process, the timing and or the location, then you may end up with heart defects. So now coming back to our ETS-1 knockout mice, this is a slide actually from a group who was studying this at the time around a few years ago as well, from Chicago, in which they could actually stain those cardiac neural crest cells in blue. So this is a normal heart, heart from a normal mouse that has the ETS-1 gene, and these blue dots just show you that this is the developing septum from a mouse embryonic heart. and you see these cardiac neural crest cells that have already made it to there, and that in the knockout mouse, and this is relatively late in heart development, there appeared to be a complete absence of those cardiac neural crest cells. So it suggested, consistent with our findings, one, that for sure ETS-1 is involved in cardiac neural crest, and that in this case, at least when you look at the later stages of heart development, for whatever reason, those cardiac neural crest cells, as in that cartoon that I just showed you, somehow are not getting out to that part of the heart where they're required to at least complete the development of the septum. So we've been very interested in knowing what are the earlier steps that give rise to that absence later on. And so don't worry too much about this slide. This is just to show you that on the top, all the blue is neural crest cells going to various structures, and in this case, these are neural crest cells migrating to the heart. And this suggests, at least qualitatively, that in the knockout mouse now that has an absence of the S1 gene, there look like there's less blue staining going to the heart. So it suggests, at least qualitatively, that there are decreased cardiac neural crest cells going to the heart. Let me show you one other thing that's a bit interesting. If you look at the facial structures of the normal mouse, you don't see any blue, but you do see blue in the facial structures of the knockout mouse, which suggests or raises the, I think, intriguing possibility that maybe instead of these neural crest cells going out to the heart where they're supposed to go, maybe they're confused and they're going out to the facial structures. That's what this result might suggest. I'm just going to skip over this slide, but basically we can just quantify to tell you in a little more precise terms exactly what we observed qualitatively. So coming back to this slide again, remember, I'm just kind of showing you specific points in time, but what we'd really like to do is get a better handle on the function of cardiac neural crest cells in the normal situation, and then how that's affected when you get rid of this gene. So one of the ways to do that is, I think, we can actually, under the microscope, identify these landmarks here for where that cardiac neural crest part of the neural tube is, and we can actually very carefully dissect that out. And what happens when you dissect out that tissue and you put it into like a petri dish, as shown in this next slide, those cardiac neural crest cells will actually migrate out. And you can actually do that in a very controlled way to begin to get a handle on whether or not that's sort of a surrogate for what might be happening in the in vitro or in vivo real life situation, if there is any effect. And so what you can, I think, appreciate here is on the left is cardiac neural crest cell tissue from a normal wild type mouse, and you can see all these cells that have just sort of migrated out radially, and we can actually quantify that. And when you look at the right, you can, I think, at least qualitatively appreciate that there are fewer cells that seem to migrate out less far. So it suggests that this would be kind of consistent with those findings before, suggesting that the loss of the ETS-1 gene is somehow affecting the total number of cells and their ability to migrate, and we can quantify that just by doing very careful measurements. Actually, and this is a very busy slide, but there are only two things I need to show you, and that is that compared to the normal mouse heart, there was clearly a statistically significantly decreased number of total cardiac neural crest cells in that system, and when you compare to the wild type, the percentage or fraction of cells that's actually able to migrate the furthest is dramatically decreased. So together, again, what this suggests is that there are decreased cells total, and those that are there have a decreased ability to migrate out. So it's kind of a double whammy that we think might be accounting for why these cells don't make it out to the developing heart to help with the normal development. So I just told you that basically in this summary slide. So still, though, we'd like to get a little bit better handle on what is actually happening in real time. And so, again, coming back to this diagram, what we're currently working on doing is to, ideally, one would like to just take a whole embryo and follow individual cardiac neural crest cells migrating during those critical times to see if there is any difference. That's a little bit difficult because the embryo is a little bit too thick to be able to track individual cells, but we can actually do something that's very close to that. And what we can do is actually take a slice I know this is a little bit graphic after lunch, of a plane through that mouse, like basically right through that plane where you get the neural tube connecting to the heart, and then you can actually, with the appropriate fluorescent markers, tag those cardiac neural crest cells and follow them with real-time imaging to see if there might be any qualitative or quantitative differences. And that's actually what we're doing right now in the lab. And this is a picture of just a normal mouse embryo. And what you can see here, just to orient you, this is kind of where the neural tube is, just like the diagram, and this is the heart. This is just a staining for all the cells, essentially, but in this panel here, what you can see is these little dots are actually cells that we've been able to specifically label that are cardiac neural crest cells, and when you look a little later on, you can actually see that there are some cells, it probably doesn't show up very well, you'll have to trust me on this, but you can actually, at a point later on in time, see that there are now some of those cells that do make it to the heart. But we want to, again, we don't want to do snapshots. We'd actually like to do real-time imaging, and that's actually what we're embarking on right now. And if you look carefully, don't blink, because this is very quick. Oops, what did I just do? Really don't blink. Try that again. So this is that slice with the neural tube up on top, right there, and the heart is down here. And then once we stain these cells, we'll be able to actually track that. So in essence, this is like having an embryo in a test tube that we can follow over real time to track the fate of those cardiac neural crest cells in the normal mice, and then of course in the mice that are missing the ETS-1 gene. So I think putting all this together strongly suggests that it may not be the cause of everything, but it certainly indicates that ETS-1 must have a pretty important role somehow in affecting cardiac neural crest cells that give rise to at least some of these heart defects. So one of the next things that we'd like to do is ask, okay, what I showed you before was when we took out the ETS-1 gene in the entire animal. But that doesn't really, that's great because at least it tells you it's got a role somewhere in that animal during development, but we'd like to be a little more sophisticated and to ask the hard question, and that is, what if you just take out the ETS-1 gene in that neural crest cell population? And we can actually do that with some genetic trickery, what we call a conditional deletion. And so don't worry about this slide in terms of the details other than to say that that's what we've been able to do. Now, in the global knockout, where we take it out everywhere from day one, those mice die right after they're born, and as I said, they all have the heart defects in that background. What we find here is that actually when we take the ETS-1 gene out just in that population of neural crest cells at the time that they're migrating, there's no effect on the heart. And what we see here, though, those first 3 mice on your left are actually the 3 mice in which we took it out just in the neural crest. And that's why they're white, because it turns out skin pigmentation is also derived from neural crest cells. So we know, and through other ways, that in fact we are taking it out in the neural crest, but they live, and their hearts are actually normal compared to their control litter mates where it's not taken out. But what was interesting is when we look a little more closely, even though they're not dying from a heart defect because their hearts are normal, I think you can appreciate here, if you look closely, some other interesting things that might be relevant to some of the problems that your children have. So the black mouse is the normal control mouse, and this is the mouse, again, where we've taken ETS-1 out just in the neural crest lineage at the time of the cardiac neural crest cell migration. And a couple of things. One I think you can see is that the spine here has this big indentation, so it's very curved. So in human patients, we call that kyphosis. If you look carefully at the appearance of the head, he has a bulging head, his ears pushed back, so posterior rotation. So it suggests that there might be some problem with the spine, again, what we call kyphosis, and something causing the head or maybe brain to be enlarged. And so we can actually do an x-ray of the spine of a mouse. And this is a normal mouse, the normal litter mate control with a nice straight spine, and what you can see here is that spine is clearly very curved. And I talked to one of my pediatric orthopedic surgical colleagues who completely concurred that that is, by definition, kyphosis in a mouse. And interestingly, no one really knows the molecular mechanisms of what causes kyphosis. There are multiple competing theories. And if you look at the brains of those mice, I told you that mouse, the one below, has what looks like a bulging head, and it turns out that mouse has a very dilated brain, dilated ventricles, what we call hydrocephalus, and it does suggest that maybe this could, at least in an indirect way, cause some of the craniofacial problems that we see. So, just to summarize, the majority of the most common heart defects that occur in Jacobsen syndrome, including hypoplastic left heart syndrome. We were able to take advantage of really the array CGH to narrow down the region, which led us to identifying the ETS-1 gene. Loss of ETS-1, we think at least, causes some of the heart defects due to its effect on that cardiac neural crest cell population, affecting both the total cell number and the ability of those cells to migrate. And that ETS-1 appears to be required for heart development prior to the establishment or prior to the time really of cardiac neural crest cell migration. And I think what's also very interesting is that, I think as I've alluded to, it's really a very compelling gene because, as I said, it was originally identified because of its role in lymphocyte development and in potentially causing leukemias and other types of malignancy. I've talked to some of my colleagues. They think it's very likely, and hopefully we'll hear about this in the next couple of hours, they think it seems very likely that this might have some implications for immunodeficiency. I think I've hopefully convinced you of its role in cardiac development in mice and maybe humans. I'll tell you a little bit more about that tomorrow when I talk about the induced pluripotent stem cell work that we're doing. And clearly it has some role, we think, in brain development, otherwise to account for why the brains are enlarged, i.e. hydrocephalus. Does that mean I need to shut up now, I guess? Okay. And certainly, as one might predict, when you impair normal neural crest cell function, you might affect melanocyte development. That's why they have that area of hypopigmentation. And these are the three people in my lab that have been wonderful to help do the real work. So I'm done. I'd be happy to take questions, and I want to thank you because I give this talk every time, an updated version of the talk to a lot of people in different areas, professionals and whatnot, but it's kind of gratifying because it all comes back to you guys. This is where it all started. So thank you, and we probably have a couple minutes. I don't know if there are any live stream questions, but I think we decided we wanted to start with the live stream questions. Are there any, Tim?
No, that's not.
Okay, just because the live stream people tend to get lost first since they can't come to me later, so if you can just hang on. Is there? They're gone? Okay, go ahead. Whatever. Jeremy, then hang on, go ahead.
Kyphosis, is that the name of it?
Kyphosis, Kyphosis. So it's an anterior or posterior curvature of the spine.
Is that something that is degenerative, or is it something that they have from birth, or is it something that can develop over?
Yeah, my understanding is like scoliosis, it tends to develop over time. I'm not an orthopedic surgeon, but yeah, it tends to be more progressive, developmental. Yeah.
We actually do have a live stream question that just came in.
Okay.
My son has a small ASD when he had tests done when he was born, but no follow-up since he was 21 months old. Does he need a follow-up?
Absolutely.
Okay.
Most small ASDs at birth close over time, but not always, so for sure should be reevaluated with an ecocardiogram.
Okay, the second question is, anything new you've seen for treating HLHS?
Well, in terms of the present case, there hasn't been a lot of dramatic changes in terms of how we manage hypoplastic left heart syndrome clinically. Again, because transplant is very unlikely to be a good option because of the lack of donor hearts. We really proceed down what's called the Norwood-Fontan stage, which is a 3-stage surgical palliation. There have been some modifications of how that approach is being done. One is the so-called hybrid approach, where you kind of delay the first stage, which has the highest mortality, and then you do it a little later on when generally babies are a little bit more stable and able to withstand the stresses of open heart surgery. Having said that, so in the clinical arena, the answer generally is there isn't a whole lot of new information in terms of how we're treating these patients. What I will say, though, is that our outcomes are definitely better, at least relative short outcomes. Here at Rady Children's, we have a 5-year survival, just based on our most recent review, I think of our last 5 years or so, of about 90%, which is as good as any center in the country. You know, a lot of that has to do with patient selection, but I know how the data were analyzed, and I think it's very objective. So and that's a manifestation, I think, of our better surgical techniques, our better anesthesia, cardioprotection, and maybe as important as anything else is our better ICU level care. Having said that, I am actually receiving today the first ever mice that have been genetically engineered that have hypoplastic left heart syndrome from a collaborator of mine. And I think that gives us a huge opportunity because as you might have deduced from what I just showed you, I've never seen really a hypoplastic left heart in the ETS-1 knockout mice, which I still think is because of probably just the genetic strain differences. But now we have a mouse that clearly has been genetically mutated to cause hypoplastic left heart syndrome, and this collaborator of mine did that for a lot of different reasons, not just to look at hypoplastic left heart syndrome, and she has graciously agreed to ship me those mice, which I just got an update last night to tell me that they're arriving today. So I think it will be very powerful to help us to understand really what the mechanisms are underlying hypoplastic left heart. And once you can do that, then you can really think out of the box about maybe better therapies. Sorry. Another live stream, hang on.
One more. Actually, I have two more. How often would we have to have our son's dextrocardia checked? He's never had any issues with it today.
So if it's just dextrocardia and an otherwise completely structurally normal heart, so that just means, if I'm just taking this at face value, there can be confusion in the nomenclature, but if it's just the heart on the right side without anything else going on, he may not need to even be followed again, if it's just that and nothing else, but you know, that's a big qualifier.
Okay, and you have one other one. Are there any effects on other areas such as developmental or cognitive of having such a heart defect? For example, ASD that has to close by itself. Does it put the strain on the body and body functions?
So the question is, does necessarily a heart defect have any impact on cognitive function? Is that the question? So in general, the answer is probably no. But there are some exceptions. So for example, it's clearly been shown that if you take all comers with hypoplastic left heart, if you look at the babies, their brains are smaller because the blood supply to their developing brain is smaller. But you know, having said that, and there are good studies that, again, taking all comers, the full scale IQ of children with hypoplastic left heart syndrome is a little bit lower, maybe 10 to 15 points lower than the norm. But I can tell you there's so much overlap between that population and normal. And I actually, the first baby that I diagnosed with hypoplastic left heart syndrome just graduated from high school a couple of weeks ago, and she is the most stunning, intelligent young lady who's actually going to go into pediatric nursing. So, you know, a lot of those studies that look at cognitive outcomes, you have to take with a grain of salt because it's old data, and what we're doing nowadays to take care, both in the operating room and afterwards, of patients with hypoplastic left heart syndrome is quite different than, say, even 18 years ago when this girl was born. So the simple answer is in terms of our studies in Jacobsen syndrome, there's no apparent higher risk factor for cognitive disability if you have a heart defect. Okay.
In discussions with your colleagues about the ETS-1 findings and the multitudes of them that you have, the missing information and how it has affected the migration of the cells and their workability, is that something that you think is going to be confirmed more in relation to the development of the leukemias, the parastrocytal issues, all of those that, you know, have the cell low quality as well as quantity?
Well, I think one thing that, if you want to look for sort of a unifying theme of what ETS-1 might be doing in different tissues, it's probably affecting or may be affecting cell proliferation or the number of cells and migration. So if it's in the heart, I just showed you what we think it's doing in the heart. For lymphocytes, it is probably affecting lymphocyte migration. If it's a form of leukemia, it might be a risk factor to affect, or some other cancers, I should say, it might be a risk factor for the metastatic, potential. So again, when you kind of step back and look at that, what's the common theme to all this? It seems to be something affecting cell number and cell migration. Does that kind of answer the question? So that's an interesting question in terms of how ETS-1 could cause kyphosis. One possibility is, again, that it has to do with that, the spinal cord is a neurocrest cell derivative, at least part of it is, as I understand it. So it may be that that's actually one of the theories, that kyphosis or scoliosis may be caused by impaired neurocrest cell function. So again, that common theme of maybe impaired migration to wherever it's supposed to go. We're all kyphosis, at least. I'm not sure if I want to say spina bifida. That's probably a different process. Okay.